Group 3, Video YouTube, Presentation.
Good day, Ma'am Moira. This is Group 3 and this is our YouTube video in Chapter 4. Entitled Nucleic Acid Purification from Plants, Animals, and Microbes in Under 30 Seconds. Introduction. The research paper authorized by Zhou et al. And published in PLOS Biology in 2017 focuses on developing a rapid and simple nucleic acid purification method for use in various applications, particularly in agricultural diagnostics. The authors address the limitation of the traditional nucleic acid extraction methods, which often required specialized equipment, trained personnel, and multiple steps, hindering the outside of laboratory settings.
Problem. The primary problem addressed in this study is need a simplified and accessible nucleic acid purification method that can be used in diverse environments, including field-based settings and resource-limited areas. The author aims to overcome the limitation of existing method by developing a technique is rapid, cost-effectiveness, and
Methodology. The study of investigate that used to cellulose-based filter paper are simple and effective materials for capturing and buffering nucleic acid. The author tests various materials and approaches, including different types of cellulose paper, membranes, and chemical treatments. They evaluate that efficiency and DNA captures and retention, as well as removal of PCR inhibitors from crude extracts, and study also explore the mechanism of DNA binding of cellulose and investigate the influence of factors like salt concentration and surface charge.
Based on their findings, the authors developed a dipstick-based nucleic acid purification method that involves three simple steps: homogenization of the sample, capture and washing of nucleic acids using the cellulose dipsticks, and elution of the purified nucleic acids into the amplification reaction mix.
Hi, I'm Brian Douglas from Bento Lab. In this video, I'm going to show you how to extract DNA for PCR using the dipstick DNA extraction method. The dipstick method is a simple, three-step protocol to extract DNA in only 30 seconds. It is cheap, quick, and it can work with a range of different samples. For this tutorial, I'm using the Bento Dipstick DNA Extraction Kit. The kit comes as a set of bottled DNA dipsticks and two solutions, an extraction buffer and a wash buffer. Let's get started. First, make sure that your tools and your workspace are clean and sterile. If you want to know more about how to do this, take a look at our guide to clean sampling practices. For each sample, you'll be using two 1.5mm tubes and a PCR tube. I've already labelled these tubes, and of course labelling is very important if you're extracting multiple samples. In this demo, my sample is a carrot which has been colonised by a mould. I've already cut a small subsample using a sterile razor blade. Make sure that your sample size is only a few cubic millimetres, otherwise it could cause the extraction or PCR to fail. The first step of the dipstick method is a cell lysis step using the extraction buffer. The buffer contains a dilute ionic detergent and salt. Aliquot a small amount of extraction buffer into the first 1.5mm tube. The exact amount depends on the size of your sample, but it's generally around 100 microliters to 200 microliters. Now, insert the subsample into the extraction buffer tube. Using a pestle, grind it for 10 seconds or until it's mostly pulverized. In combination with the grinding of the sample, the buffer will rupture the cell membranes and release the DNA. Finally, top up with more extraction buffer to a total of 500 microliters and close the lid. Next, aliquot 1 milliliter of the wash buffer into the second 1.5mm tube and close the lid. Finally, prepare your PCR mix and aliquot it into the 0.2mm PCR tube. Now for the dipstick and wash steps. The dipstick has a cellulose binding zone at one end. This is where the DNA will bind to the dipstick. Capture the DNA by dipping this end in the extraction buffer 3 times. You want the binding zone to be completely soaked. Then gently dip the dipstick into the tuber wash buffer 5 times. When you remove the dipstick, check that there are no large droplets of buffer on the dipstick. If there are, gently touch the dipstick to the edge of the tube to remove the droplets. Now we release the DNA into our PCR solution. Dip the dipstick into the PCR reaction mixture between 3 and 15 times, depending on how concentrated your sample is. Push the dipstick into the bottom of the tube so that the dipstick bends. This helps the liquid move through the paper and helps increase the amount of DNA released. After dipping, wipe the dipstick on the edge of the PCR tube to make sure that there's no large droplets of PCR reaction mix left on the dipstick. The PCR reaction is now ready to go. If you're not planning to perform PCR straight away, you also have the option of storing the DNA in a small amount of TE buffer. A 1.5mm tube is suitable for this. Simply dip the dipstick a couple of times into the solution and then leave the dipstick in the tube as you close the lid. The standard extraction buffer is suitable for a wide range of different tissue types. In some cases, such as for particular animal or fungal samples, it might be useful to use a more concentrated version of the extraction buffer in order to increase the effectiveness of the method.
Finding and result. The study reveals that untreated cellulose-based paper, such as Whitman, number 1, felt paper can effectively capture and retain nucleic acid within seconds. Well, removing contaminant during a single washing step. The author demonstrates the successful application of this method to a wide range of plant, animals, and microbial samples, including blood, leaves from manure tree, and various agricultural practice. Therefore, demonstrate the ability of this method to clarify DNA from plant and animals pathogen, including bacteria and fungi, and the RNA from plant virus. The dipstick-based method provides a significant faster and cheaper than commercial available rapid nucleic acid purification system, which achieve comparable sensitivity and efficiency.
Here's the application in agriculture. This research has significant implication for agriculture. The developed method offers a simple, rapid, and cost-effective solution for nucleic acid extraction in various agricultural settings, including: first, disease diagnostic. The method can be used to rapidly detect plant and animal pathogens, facilitating early diagnosis and intervention. Second, genetic analysis. The method can be applied to analyze genetic diversity, identify disease-resistant genes, and select desirable traits.
Crop improvement. The method can be used for marker-assisted selection, allowing the breeder to select for desired traits more efficient. Field-based testing. The method's simplicity and portability make it suitable for on-site diagnostic, enabling rapid disease detection and monitoring in the field.